Hello friends, good evening. Welcome to Sam Biology class. Friends, today we shall discuss about various theories that explain the mechanism of crossing over. One of the theory is classical theory, which was proposed by Carl Sachs in 1932. According to this theory, formation of chiasmata precedes the act of genetic crossing over. That means, first of all, formation of chiasmata takes place, then it is followed by crossing over. So, chiasmata are the cause but not the result. According to this theory, it is not necessary that there is one-to-one relationship between chiasmata formation and crossing over. So, chiasmata represent the accidental physical crossing between non-sister chromatid, and the formation of chiasmata may or may not lead to the breakage and rejoining of non-sister chromatids. But whenever crossing over takes place, it occurs due to the strain imposed by the formation of chiasmata. Another theory regarding the mechanism of crossing over is chiasmata type theory, which was proposed by F.A. Janssen in 1909. Later, this theory was fully developed by other workers such as J. Belling and C.D. Darlington. This theory is opposed to classical theory. According to this theory, crossing over precedes the formation of chiasmata. That means, first crossing over takes place and then as a result, chiasmata are formed. So, there is one-to-one relationship between crossing over and formation of chiasmata. According to this theory, as opposed to classical theory, crossing over takes place because of breakage and rejoining of non-sister chromatids. Darlington's strain theory explains the mechanism of crossing over in a different way. According to this theory, in zygoting, the chromosomes of homologous pair are coiled around each other in a particular direction. Similarly, the chromatids in each homologous chromosome are also coiled around one another but in an opposite direction. As the chromosomes start separating, the unraveling of coilings in opposite direction create torsion, which exerts strain on the chromatids. As a result, the chromatids are broken at contact points, after which rejoining of these broken chromatids create new combinations of genes. Another theory of crossing over is copy choice theory, which was proposed by John Belling in 1928. Belling visualized genes as bits described as chromomeres. These chromomeres are connected by interchromomeric regions. According to this theory, during duplication of chromosomes, the chromomeres are duplicated fast, and the newly formed chromomeres are tightly just opposed to the older ones. When the interchromomeric regions are synthesized, it may switch from one chromomere, which is synthesized from one chromosome, to another chromomere, which is synthesized from the other homologous chromosome. The switching of interchromomeric regions from one chromomere to another chromomere create new combinations of genes. Another model explaining the mechanism of crossing over is switch model, which was proposed by Joshua Lederberg in 1955 in order to explain recombination in microbial system. This model is a modified version of Belling's hypothesis. According to this model, the newly synthesized chromatid is derived by copying one homologous chromosome up to a certain distance, then it switches on to the other homologous chromosome for copying remaining part. The copy choice model is not adequate for two main reasons. Number one, this model assumes that DNA replication is conservative, but experimental evidences show that DNA replication is always semi-conservative. Number two, according to copy choice theory in meiosis, whenever multiple crossover takes place, only two chromatids are involved. But experimental evidences show that during multiple crossover, 3 or even 4 chromatids may be involved. Another concept is breakage and exchange theory, which was suggested by Stern and Hutta in 1969. According to this theory, breakage of non-sister chromatid takes place by the action of enzyme nuclease. After that, the broken chromatids are rejoined by using the enzyme ligase. Because of this, new combinations of genes are formed. Friends, genetic exchange at appropriate positions along two chromosomes with substantial DNA sequence homology is referred to as normal or homologous recombination. It means homologous recombination takes place between two chromosomes which have substantial sequence similarity. Several models attempt to explain homologous recombination. All such models share certain features in common. First, they are all based on proposals put forth independently by Robin Holliday and Harold L. K. Whitehouse. Second, they all depend on complementarity between DNA strands. And third, all such models involve a series of enzymatic processes in order to achieve genetic recombination. Today we shall discuss about one such model which explains the molecular mechanism of crossing over and homologous recombination. This model, which is known as Holliday model, begins with pairing between two DNA duplexes in each of which an endonuclease introduces single-stranded nick at identical position. This is followed by strand displacement in which the internal strand endings produced by these cuts are displaced and subsequently paired with complements on opposite duplex. Strand displacement is followed by ligation. In this step, the loose ends are sealed by the action of the enzyme DNA ligase. The process of ligation creates heteroduplexes which are called as heteroduplex DNA molecules. The heteroduplex DNA molecules created by ligation are held together by crossbreeds-like structure. The position of the crossbreeds move down the chromosome by process called as branch migration. Branch migration occurs as a result of zipper-like action as hydrogen bonds are broken and reformed between complementary bases of displaced strands of each duplex. Branch migration increases the length of heteroduplex DNA on each homolog. If the heteroduplexes are bent and its bottom portion rotated by 180 degrees as shown in this figure, an intermediate planar structure is obtained. The structure is known as chi-form or Holliday structure. In the next step, the enzyme endonuclease introduces single-stranded nicks in two strands of DNA on opposite homolog, which were previously not involved in exchange. Nicking is followed by ligation, in which the enzyme ligase seal the loose ends that result in the formation of two recombinant duplexes. Similarly, the enzyme endonuclease can also create single-stranded nicks in the two remaining strands of DNA, after which ligation occurs. As a result, two more recombinant duplexes are formed. So friends, by this process, the arrangement of alleles is altered, and as a result, genetic recombination takes place. In addition, other models have also been proposed to explain the mechanism of homologous recombination. All these models involve the creation of double-stranded break in one of the two duplexes of DNA. So friends, this is all about the molecular mechanism of crossing over and genetic recombination. For more updates, please subscribe my YouTube channel, Sam Biology Class.